the last topic of cell and molecular biology, the biotechnology and genomics. Let us start with DNA cloning. What is DNA cloning? DNA cloning is a molecular biology process of making identical copies of DNA segments located in the chromosomes that contain the genetic code of advanced organisms. The aim of DNA cloning is to produce the target DNA sequences themselves or to produce the proteins encoded in the target sequences. There are two methods used in DNA cloning. First is the Plasmid vector method and the polymerase chain reaction. In the Plasmid vector method, DNA strands are cut using restriction enzymes to produce DNA fragments, and the resulting segments are inserted in cloning vectors called plasmids for further duplication. The plasmids are placed in bacterial cells that then produce DNA copies or encoded proteins. While in polymerase chain reaction, the segment of DNA strands to be duplicated is marked with enzymes called primers. A polymerase enzyme makes the copies of the marked part of DNA strand. This method doesn't use restriction enzymes and can produce cloned DNA from small samples. Sometimes, the two DNA technology methods are used together to incorporate the best features of each in an overall reaction. Plasmid vector method. The vector of the method refers to the plasmid used to hold the target DNA segment to be cloned. Plasmids are small circular strands of non-chromosomal DNA found in organisms including bacteria and viruses. Bacterial plasmids are the vector used for inserting the target DNA segment into bacterial cells for further duplication. Number 2. Cutting the target DNA with restriction enzymes. Restriction enzymes are selected to look for the DNA code at the beginning and end of target sequences. When the restriction enzymes find a special coded sequence or base pairs called restriction sites, they attach themselves to the DNA at the location and wind themselves around the DNA molecule, severing the strand. The cut DNA segments containing the target sequences are now available for duplication. The third process is choosing the plasmid vector and inserting the target DNA. A suitable plasmid ideally contains the same DNA coding sequences as the DNA strand from which the target DNA was cut. The circular DNA strand of the plasmid is cut with the same restriction enzymes as were used for cutting the target DNA. A DNA ligase enzyme is used to promote DNA segment linking, and the ends on the target DNA segment link up with the cut ends of the plasmid DNA. The target DNA are now forms part of the circular plasmid DNA strand. Number 4. Inserting the plasmid into a bacterial cell. Once the plasmid contains the DNA sequence to be cloned, the actual cloning can take place using a process called bacterial transformation. The plasmid are inserted into a bacterial cell such as E. coli, and the cells with the new DNA segments will start producing copies and the corresponding proteins. In bacterial transformation, the host cells and plasmids are incubated together at the body temperature for 12 hours. The cells absorb some of the plasmids and treat them as their own plasmid DNA. Lastly, harvesting the cloned DNA and proteins. Most plasmids used for DNA cloning have antibiotic resistance genes incorporated in their DNA. As the bacterial cells absorb the new plasmid, they become resistant to antibiotics. When the culture is treated with antibiotics, only those cells that have absorbed the new plasmid survive. The result is pure culture of bacterial cells with cloned DNA. That DNA then be harvested or the corresponding protein can be produced. We are done with the process of the plasmid vector method. Now let us proceed to polymerase chain reaction method or the PCR method. The PCR method is simpler and copies existing DNA in place. It doesn't require cutting with restriction enzymes or inserting plasmid DNA sequences. This makes it especially suitable for cloning DNA samples with a limited number of DNA strands. While the method can clone DNA, it can't be used for production of the corresponding protein. The first process in the PCR method is uncoiling the DNA strands. DNA in chromosomes is tightly coiled in a double helix structure. Heating the DNA to 96 degrees Celsius in a process called denaturation makes the DNA molecule uncoil and separate in two strands. This separation is required because only a single strand of DNA can be cloned at one time. Selecting the primers. As a plasmid vector DNA cloning, the DNA sequences to be cloned have to be identified with special emphasis on the beginnings and ends of the DNA segments. Primers are enzymes that attach to specific DNA code sequences, and they have to be selected to mark the target DNA segments. The right primers will attach to the DNA molecule sequences to mark the beginnings and ends of target segments. Number 3. Annealing the reaction to bind with the primers. Cooling the reaction down to 55 degrees Celsius is called annealing. As the reaction cools, the primers are activated and attach themselves to the DNA strand at each end of the target DNA segment. The primers act only as markers and the DNA strand does not have to be cut. Number 4. Producing identical copies of the target DNA segment. In the process called extension, the heat-sensitive TAQ polymerase enzyme is added to the reaction. The reaction is then heated to 72 degrees Celsius, activating the enzyme. The active DNA polymerase enzyme binds to the primers and copies the DNA sequence between them. The initial DNA sequencing and cloning process is complete. Number 5. Increasing the yield of cloned DNA. The initial annealing and extension process creates relatively few copies of the available DNA strand segments. To increase the yield through additional DNA replication, the reaction is cooled again to reactivate the primers and let them bind to the other DNA strand. We can also use the plasmid vector method and the PCR DNA cloning methods together. The plasmid vector method relies on an ample initial supply of DNA to cut and insert into plasmids. Too little original DNA results in fewer plasmids and a slow start to clone DNA production, while in the PCR method can produce a large quantity of DNA from few original strands, but because the DNA is not implanted in the bacterial cell, protein production is not possible. Then the PCR method products are used with the plasmid vector to implant the produced DNA into bacterial cells that will produce the desired protein. Here are some applications of DNA cloning. Number 1, the gene for human insulin is cloned in bacteria that then produce the insulin used by diabetics. Number 2, Tissue plasminogen activator is produced from cloned DNA and used to help prevent blood clots. Number 3, human growth hormone can be produced and administered to people who can produce it themselves. And number 4, biotechnology also uses gene cloning in agriculture to create new characteristics in plants and animals or enhance existing characteristics. As more genes are cloned, the number of possible uses